Hello everyone, my name is Tushar. I am from Kirodimal College, BSc Chemistry Honours 3rd year. Today I am going to explain three major topics of biochemistry. First one is the silent feature of active sites of enzymes. Second one, transcription. Third one, translations. These three topics basically shows how our cell run, how reactions happen in our biological system, and how genes convert into proteins. So, in the first slide, let's introduce what is an enzyme. What is an enzyme? Enzymes and gene expressions are two central concepts in our biochemistry. Enzymes act as a biocatalyst which promotes a controlled biochemical reaction inside cell. Their activity depends mainly on the active sites. And now a question raises that what is an active site? So active sites is a site, a special region where the reaction take place. So what is an we will discuss more about on the active sites of enzymes. The active site is a small, specially shaped part of an enzymes where the substrate bind and the reaction occurs. It is usually a three-dimensional pocket or groove formed by the folding of enzymes polypeptide chain. So only certain molecule can fit into this region, make enzymes action highly specific. As you can see, here is the diagram in the slide. So it is an enzyme, it is substrate, it is binding site and catalytic site. So what is the binding site and catalytic site, we will surely discuss in later. So silent features. In silent features we will discuss like first, three-dimensional structure formed by a precise folding of amino acids far apart in a primary structure. It creates a unique shape essential for the substrate binding. Specificity: enzymes recognize only specific substrate based on the lock and key model or induced fit model. We will what are the lock and key model and what are the induced fit model, we will discuss in later. Microenvironment: active sites may have a different pH or polarity than the rest of the cell. So it provides ideal conditions of the catalyst. Weak interactions: in interactions, substrate bind through hydrogen bonds, van der Waals forces or ionic interactions. These interactions are temporary bonds helping to quick binding or release of a bond. Induced fit. Active sites change shape slightly after the substrate binding. So when the substrate come onto the active sites, the active sites change slightly for the binding. Improves alignments of catabolic group of a reaction completion. Catalytic residue: specific amino acids directly involved in a breaking of forming chemical bonds responsible for lowering of activation energy. So as more example like serine act as an serine protease, histidine act as an acid-base catalyst, cysteine forms a covalent intermediase. So these are some examples of the catalytic residues. So next, type of active sites. That first we will discuss the binding site. An area where a substrate is physically held in place or helps correct orientation of the substrate. Binding site is a site where the substrate can hold together and the orientation of the correct orientation of the substrate. So next point, catalytic site performs an actual chemical reactions, breaking bonds, forming bonds and transferring groups. Together these two creates a full functional active site. Next we will discuss about the lock and key model. Lock and key model. It was proposed by the M.L. Fisher in 1894. This model suggests the active sites is preformed as a rigid structure completely to the substrate, like a key fitting a lock. Only the correct substrate shape can enter and binds effectively. Like we know the mechanism of the lock. So Emil Fisher think like that, and he gave a model of that here the enzyme is a lock and substrate is a key. So log opens when a certain specific key unlocks that. Next one. So next we will move that the lock and key model. According to the lock and key hypothesis, the specificity of an enzymes, the lock, and its substrate, the key, come from their geometrical complementary shapes. In an enzyme's catalytic reaction, the enzyme and the substrate combine to form an enzyme-substrate complexes. So when the enzyme and substrate mix together, then the enzyme-substrate complex form. Formation of an enzyme-substrate complexes often introduce a conformation changes in the enzymes called induced fit. That allows it to bind the substrate more effectively. The place where a substrate binds to an enzyme and where the reaction take place is called active sites. So as in the I'll show you. So in this diagram we can say that the binding sites and enzymes and substrate here that is known as the active sites. Reaction catalyzed by enzymes are stereospecific because enzymes are chiral in nature. A compound that can negatively alter the activity of enzymes is called an inhibitor. Next point, induced fit model. Induced fit model, it was proposed by the Daniel Koshland in 1958. He stated that the active sites are flexible in nature, so the substrate binding introduce conformational changes that optimize the catalytic geometry, creating the perfect fit only upon the substrate engagement. As you can see in the model, so in lock and key model, the both enzymes and substrate are fixed, but in induced fit model, he said that the enzymes are flexible in nature, so substrate can induce itself. The shape of the active sites and the substrate must be still be responsibly similar, but once bound, the shape of the active sites is different from the active sites in the unbounded enzymes. Means when the sites are induced, so it makes a complex enzyme product complexes. A well characteristic enzymes of the site induced fit model seen by in the binding of the glucose to form a hexokinase. Factor affecting active site functions. First one is the temperature. Temperature on high temperature may denature the enzymes, destroying the active sites. Low temperature slow down the movement, which reduce the enzyme activity. pH, you know our body is a very much pH sensitive, so changes in pH can alter the charge and shape of the amino acids in the active sites. Substrate concentration, low substrate concentration, low reaction. High substrate concentration, active sites become saturated. So it is very much important to maintain the substrate concentration in the body. Or a inhibitor, competitive inhibitor blocks the active sites. Non-competitive inhibitor changes enzyme shape indirectly. Cofactors and coenzymes. Some enzymes require metal ion or vitamin to activate the active sites. Now we will come on the gene expression. So what is the gene expression? Gene expression is the process through which the information stored in the DNA is used to produce a functional protein. So here we will discuss about the two main topics, transcription and translation. So you can see in the picture, this is the DNA. For after the transcription, it converts into the mRNA. The full form is the messenger ribose nucleic acid. After the translation of the mRNA, the protein chain comes. So first, transcription. Transcription is the synthesis of the messenger RNA from a DNA template. It carried out by the enzyme RNA polymerase. In eukaryotic, it occurs inside the nucleus. Produce a complementary RNA strands based on a DNA sequence like AU-GC pairing. You can see in the diagram also. Steps of transcription. Initiation. RNA polymerase binds to the promoter region. DNA unbinds to expose the template strands. Elongation. RNA polymerase move along DNA and RNA nucleosides like AUGC. The mRNA strands grow into 5 and to 3 direction. Termination. A termination signal stops transcription. mRNA is released. When the transcription stops, mRNA releases. mRNA processes in eukaryotic. 5' caps protects mRNA. Poly A tail stabilize mRNA. Splicing removes introns, joins exons. Characteristics of transcription. Use DNA as a template. Produces single strand mRNA. Occurs before translations. Provides codons that will later guide protein synthesis. Translation. Translation is the process where the ribosomes reads the mRNA sequences and convert into the polypeptide chain, like that is our protein. It happens in the cytoplasm at ribosomes. It uses codon. What is the codon? I'm pretty much sure you will confused in the what is the codon. So codon 3-letter RNA sequences to add correct amino acids. On its needs for translation. mRNA contains codons that specify amino acids. tRNA, that is a transfer RNA, carried amino acids to the ribosome. Each has anticodon. So reverse of the codon. Ribosomes, workbenches of the protein synthesis, have large and small subunits. Amino acids, building block of the proteins. Enzymes and factor helps in initiation, elongation and termination. ATP, GTP provides energy for the processes. Steps of the translation. Initiation. Ribosomes binds to mRNA at the start codon AUG. First tRNA brings methionine, as you can see in this diagram also. Elongation. Ribosomes moves along mRNA codons by codon. New tRNAs bring amino acids. Amino acids join through peptide bonds. Termination. Stop codon AUAA, UAG, UGA reaches. Polypeptide chain is released. Ribosome detached from the mRNA. So the final conclusion is that the active sites of enzymes is highly specialized region where all biochemical magic happens. Transcription converts DNA instruction into mRNA. Translation uses these instructions to build proteins. These three processes together shows the efficiency, intelligence and perfect coordination inside our cell. Thank you. Presented by Tushar.